So, hello and welcome everyone. Our speaker for today's talk is Dr. Samira Ghorbani. Dr. Ghorbani is a postdoctoral fellow at the University of Calgary. She graduated in Immunology from Tehran University of Medical Sciences in Iran. Her PhD thesis was focused on the role of microRNAs in immunopathogenesis of MS. Her interest in the MS field has led her to pursue a postdoctoral fellowship with Professor Young at the University of Calgary, where she investigates the role of different extracellular matrix members on remyelination in MS. And today she's going to talk about immunology in MS. So Samira, looking forward to your presentation.
Thank you so much. Hello, everyone. Good morning. Thank you, Nitu, for introduction. As you know, today we are going to talk about the immunology of multiple sclerosis. Because the target audience is summer student, and I know that some of you are not familiar with immunology. So I will start with first review some basics of immune systems. Then we'll talk about the autoimmunity and MS as an autoimmune disease. Next, we'll see what kind of immune cells are involved in MS. And then our next subject will be about the initial trigger in MS and how immune responses against CNS evolve. Finally, we will review the immunopathogenesis of MS at early and also late stages. As Dr. Kuiper explained it nicely last week, innate and adaptive immune responses are components of the host defense. Innate immunity are the initial defense against infection, and it responds so rapidly. But on the other hand, adaptive immune responses develop later. Innate immunity is a specific for common structure in microbe and cannot distinguish the differences between microbes. But adaptive immunity is able to distinguish different substances, which is called specificity. And also, adaptive immunity is able to respond more robust to repeated exposure, which is known as memory. These are two important features of adaptive immunity. They are connection between the innate and adaptive immunity. The innate immune responses are needed for the development of adaptive immunity. On the other hand, adaptive immune responses often enhance the innate immunity. The innate immunity consists of different cell types such as phagocyte, neutrophil, macrophage, monocyte, or dendritic cell, and also NK cells. And the unique components of the adaptive immunity are called lymphocytes. Lymphocyte consists of different population. B lymphocyte recognize soluble antigens and develop into antibody-secreting cells. Helper T cells or CD4 positive T cells recognize antigens on the surface of cells and secrete cytokine which activate cytotoxic T lymphocyte, which activate other immune cells such as macrophages, and these cytokines can enhance inflammation, and also they promote the proliferation of the lymphocytes and other immune cells. We also have CD8 positive T cells or cytotoxic T cells, which they recognize the antigen on the infected cells and they can kill these cells. There are also some regulatory T lymphocyte or B lymphocyte that they are able to suppress and prevent immune responses, and they can control the inflammation. But as you know, this is so simplified. And there are several subtypes for each class. For example, T helper 1, I am not sure if you can see my pointer, but for example, T helper 1, which secrete the IF and gamma, and T helper 17, that they can secrete the IL-17. IL-17 is the most important cytokine produced by these cells. T helper 1s are more important in the infection, in the intracellular infection, and IF and gamma can promote the phagocytosis in macrophages. but TL-17 are more important in defense against the extracellular bacteria or fungi. And both these cells, TL-1 and TL-17, are very important in the autoimmune diseases, and they can mediate the tissue damage in these kind of disorder. There are also some kind of cells that they can produce both IFN gamma and IL-17. On the other hand, some naive CD4 positive T cells can develop into Th2, which these cells secrete IL-4 and IL-5, which are important in the defense against helminths. There are also some regulatory phenotypes of helper T cells, such as FOXP3 positive Treg or TR1. These cells, actually, they secrete some immunosuppressive cytokines such as IL-10, and also they are able to express some molecules on the membrane, which can mediate the suppression by cell contact to other immune cells. For the initiation of the adaptive immune responses, antigen needs to be captured and displayed to lymphocyte. The cells that do this role are called antigen presenting cells, or APCs. As you can see here on the upper left, the T cells in red and dendritic cell in blue, they are interacting together and there is a synapse between them. After T cell generation in the thymus, they go to the lymph node and spleen and they patrol the whole body. They get activated if they detect their specific antigen on the surface of APCs. Here in this video, we can see that the T cells are patrolling the lymph node, and if they detect their specific antigen on the surface of APC, they get activated and they secrete some cytokine that this cytokine can recruit other lymphocytes to the site. And also this cytokine can promote the proliferation of these lymphocytes. On the left also there is a video from the lymph node. Specific T cells are in red and dendritic cell in green. You can see they are going through the lymph node, and when they detect the antigens on the dendritic cells, they reduce their speed and they increase DC contact so they can make a synapse and they can get activated. So T cell can recognize the specific antigen with binding to the MHC peptide complex on the surface of APCs. But this binding alone is not sufficient to trigger T cell activation, as you can see here in panel B. So other co-stimulatory molecules are needed, which we call them second signal. The most powerful second signal is interaction between CD28 on T cells to CD80 or 86 on APCs. APCs do not usually express these co-stimulatory molecules under steady state. They need to get activated throughout their innate receptor to express these co-stimulatory molecules. So initial inflammation is needed to trigger T cell activation. When T cells are activated, then we call them effector T cells, and they do not need the second signal anymore, and they can get reactivated by just detecting the antigen on the surface of APCs. Another features of lymphocyte is their receptor diversity. It means that one person has millions of different antigen receptors. In other words, we can say naive T cell pool can detect millions of antigens. But how lymphocyte can distinguish self-antigen from foreign antigens? During lymphocyte development, there are mechanisms which remove these self-reactive cells. This process is called tolerance. Immature lymphocytes, which are specific for self-antigen, may encounter these antigens during their development in the thymus or bone marrow in case of B cells. And they can get deleted, or they can change their specificity, their receptor, or they may develop into regulatory lymphocyte. But this is a central tolerance occurring in the thymus or bone marrow. This is not perfect, and some self-reactive lymphocytes may mature and enter peripheral tissues. There are also peripheral mechanisms that keep these autoreactive cells in check and keep them quiet. They may be inactivated or deleted when they see their specific antigen in periphery, or they may get suppressed by other regulatory lymphocytes. So we have central and peripheral tolerance. But regardless of tolerance, autoimmunity occurs. MS is also considered as an autoimmune disease, which immune cells attack the CNS and cause the damage. So the question here is that how self-tolerance fails and how self-reactive lymphocytes are activated. So as you know, the answer to this question is a major challenge in immunology. And there is no exact and clear answer to these questions. But there is a proposed model of MS saying that the various genetic loci may increase susceptibility to autoimmunity. So these genes probably influence the self-tolerance. And on the other hand, we have environmental triggers such as infection or other inflammatory stimuli, which promote the influx of lymphocyte into the tissue. And finally, activation of self-reactive T cells, and which result in the tissue injury. So the cause of MS is unclear and remains elusive. Generally, MS is thought to happen in genetically susceptible individuals with environmental factors affecting the disease. So genetics account for 30% of overall disease risk. More than 200 genetic regions have been identified to be related with MS. Most of these genes are immune genes, and some of these genes are from the HLA or MHC family. So it is not surprising because we know that MHC has a role during the tolerance. They can present antigen to the T cell during their development, so they can affect the specificity of T cells. There are also non-HLA genes related to MS, but they are still immune genes, such as cytokines IL-2, IF, and gamma. And in addition to genetic factor, we have environmental factors such as UV, vitamin D deficiency, smoking, EBV infection, and also microbiome, which these factors can contribute to the disease development. MS is known as an autoimmune disease, but we don't know how correct it is. The thing that we are sure about is that MS is a neuroinflammatory disease, which is associated with neurodegeneration. So these days, MS is categorized as a neuroinflammatory degenerative disorder. There are two important features in MS, neural cell death and inflammation, which they are in a positive feedback loop, and they can enhance each other. The pathological hallmark of MS is the formation of the demyelinated area in the brain, in the bottom, as you see, and also in the spinal cord. The lesions, these demyelinated areas are called plaque or lesions and show a loss of myelin sheath and oligodendrocyte. They have been seen in both gray matter in the cortex and also in the white matter. But what happened in these lesions? Lesions are thought to be caused by infiltration of immune cells, including T cells, B cells, and myeloid cells. And these cells work together and they attack the myelin sheath and cause injury. The most common type of the early or acute lesion in the middle panel, as you can see, these are the acute lesions. So macrophage dominate the infiltrate, followed by CD8 positive T cells, and also there are numbers of CD4 T cells and a few number of B cells and plasma cells. When disease continues and it progresses during the chronic phase and later stages, infiltration decrease and macroglia and astrocyte activation are more prominent. And most of the lesions during the acute phase in the progressive MS show a small amount of inflammation there are some inflammation, particularly at the edge of the lesion. But there are still myelin damage. This is continuing process. In some lesions, we can see that inflammation is completely resolved without remyelination. So we call these lesions chronic inactive lesion. But also there are some lesions that we can detect some amount of remyelination. Here we can see the MS lesions, and in panel A, you can see that they are easily detectable. They are a bit darker compared to their surrounding white matter. Or in panel B and C, they are less blue because of less myelin content. We can detect both T cells and B cell in MS prevascular infiltrate. CD3, as you can in the bottom, first panel, is a marker for both helper T cells and cytotoxic T cells, and there are a lot of T cells in the infiltrate. There are some of them are CD4 positive helper T cells, but CD8, you can see that CD8 are the prominent lymphocyte class among the infiltrate. And also there are a few of the B cells and plasma cells in the prevascular infiltrate or parenchyma of MS lesions. The main part of our knowledge about MS is from animal models. You know that there are different animal models, but most common and best characterized model for a T cell mediated autoimmune neuroinflammation is EAE. So we induce EAE by immunizing mice with the CNS antigens, such as myelin oligodendrocyte glycoprotein. We inject antigen lined with adjuvant, which is a killed mycobacterium. This adjuvant is necessary to activate innate immune response, and also it is necessary to induce the initial inflammation, and this inflammation is needed for a strong T cell response. After 1 to 2 weeks after immunization, animal develop encephalomyelitis, which is characterized by prevascular infiltrate composed of lymphocytes and macrophages. And finally, there is a tissue damage in spinal cord. There is abundant evidence that in mice, EAE is caused by activated T helper 1 and T helper 17 cells. As you can see in the last panel on the top, the EAE disease can be also transferred from naive animal with transferring T cells from diseased animal to control animals. So it is the adaptive model of EAE and emphasize on the role of T cells in EAE pathogenesis. On the bottom, you can see the image of a spinal cord from EAE mice. Myelin is shown in green and immune cells in red. It shows several demyelinated areas in the white matter. As you can see in the image with higher magnification, demyelinated is shown, it can be detected by loss of myelin, which is the loss of the green signal. And this demyelinated area is full of immune cells. T cells are not the only cells seen in EAE. B cells are also present in meningeal compartment. As you see in the left image, B cells are shown in red, and you can see there are a lot in the meningeal compartment of the EAE mice. In this slide, we can appreciate the change in the composition of immune cells within the CNS. Under steady state, macroglia, as you can are the main immune cells of CNS, and there are also some border-associated macrophages shown in yellow. They are meningeal macrophages, for example, and also a few T cells. T cells are shown in blue. But during inflammation in EAE, there is a huge increase in number of T cells, as you can see here in blue. And also there is expansion of the dendritic cell population and also blood-derived macrophages. Other obstacles. Yes.
You can see your cursor if you're pointing.
Yeah, that's the problem that we talked about. I don't know. That's my laptop problem.
I just wanted to let you know. Okay.
Yeah, sorry about that. I'm trying to guide you to the places I'm talking about, but sorry about that. So, and also there are other populations such as B cells and also neutrophils that are increased during the EAE. In this video, we can see the infiltration of effector activated T cells in adaptive EAE model. Autoreactive T cells are shown in green in the left video. And so they injected in a T cell deficient mice. So they enter and also exit the parenchyma, as you can see, using the meningeal vessels. And in the right panel, we can see the activation states, which is shown in red. We can see when they enter the parenchyma, they can get reactivated. And in this video, we can see the direct and close interaction between T cells and axons in EAE lesions. So this interaction finally causes the axonal damage and tissue damage in EAE. Sorry. In MS patients, helper T cells are found within CNS lesions and in the CSF. According to EAE model, there are two important T helper subtypes in driving the disease, T helper 1 and T helper 17. So they produce some cytokines, which these cytokines enhance inflammation. And also they can activate, macroglia, macrophages, and astrocyte, and make these cells to produce some other cytokines and produce reactive oxygen species, and also improve the antigen presenting function of these cells. T helper 17 can also regulate B cell activation and plasma cell generation through the IL-1 beta production. But T helper 17 population is so heterogeneous. There are some pathogenic and non-pathogenic T helper 17 cells, and also IL-17, which is produced by these cells, has many different functions, and its receptor is widely expressed on different cell types. It has been reported that IL-17 also has a role in blood-brain barrier damage and also has a role in tissue repair. And in MS, some T cells have been detected that they can produce both IFN gamma and IL-17. Anti-IL-17 therapy is successful in decreasing lesion formation, but on the other hand, it increases infections. So this therapy failed in clinical trials. GM-CSF is a cytokine that can be produced by both cells. It hasn't been shown here, but it is a very important cytokine during the MS pathogenesis. And it has been shown that it's necessary for EA development. Even it is more important than IL-17 and IFN gamma. And recently, a new subset of T helper cells has been discovered in MS, which is defined by GM-CSF production. GM-CSF can increase the immune activation and also infiltration, and anti-GM-CSF therapy is now in clinical trial. Another cell type which is important in MS pathogenesis is CD8 positive T cells or cytotoxic T cells. They make up for the majority of T cells in the prevascular infiltrate and also at the edge of MS lesion, but not EAE. These cells are able to kill the target cells directly. They can produce the produce the IL-17 and IFN gamma, which are important, can be in the periphery or in the CNS by regulatory T cells. And for example, Treg cells or regulatory B cells has been reported in MS patient. Also, effector cells from patients are more resistant to suppressive mechanism of regulatory lymphocytes. Another thing that I want to mention here is the plasticity between Treg and TLP1 or 17. They can switch to each other, and some therapies which are successful in MS probably can switch the balance between regulatory T cells and Th17 cells. So they can increase the population of regulatory T cells. So in this way, they can affect the disease. Also, NK cells show regulatory effects in MS. Reduced numbers of NK cells is associated with relapse, and NK depletion worsen, actually make the EAE worse. But what about B cells? In recent years, more attention has been paid to B cells. It's because of the dramatic effect of B cell targeting therapies such as rituximab and ocrelizumab. These therapies target and delete B cells. And so they have a good effect on reducing relapses and also some effect on the progression of the multiple sclerosis. So they are showing some role for B cells in MS. But what are the function of B cells? B cells can produce autoantibodies in the periphery and CNS. It has been shown that antibodies, but probably these antibodies are not very important because the therapies that target B cells but not plasma cells are effective in MS. And if we target the plasma cells, only the plasma cells, actually we make the situation worse. Another function of B cells is the antigen presentation. They can present antigen and the MHC2, and they can actually enhance the inflammatory condition. This function of B cells is considered as their central role in MS pathology, because in mice that B cells do not express MHC2, EAE cannot be developed. Another function of B cell, which is important, is secreting pro-inflammatory cytokines such as TNF-alpha, GM-CSF, and IL-6, and these cytokines promote inflammation. And another proposed mechanism for B cell therapies actually is reducing the GM-CSF production, because these therapies can delete B cells, so GM-CSF can be reduced. And also there are some regulatory B cells that we talked about them before. So these are all kind of immune cells, lymphocytes, that have been detected in patients and isolated from the blood and also CNS. But the question here is that how these cells are activated in patients. What is the initial triggers? So there are two different theories regarding pathogenesis of MS, peripheral, and which is the CNS extrinsic model or outside in model, and another model is CNS intrinsic model or in out model. In the periphery model, autoreactive T cells are activated in periphery because of molecular mimicry or other factors. And then they traffic to the CNS along with activated immune B cells and macrophages. In other model, intrinsic events initiate disease development and inflammation. So infiltration of autoreactive T cells occur as a secondary phenomenon. For example, unknown viral infection or some processes, some neurodegenerative processes such as oxidative stress or oligodendrocyte stress may be the cause of for this model. Peripheral model is more accepted because there are lots of evidence. So we talked about the immune genes which are associated, and among these genes there is overlap between MS and other autoimmune diseases, but there is no overlap between MS and neurodegenerative disease. On the other hand, there is no main environmental factor affecting the MS, and also there is no clear autoantigen for MS, which support the intrinsic model. If we consider MS as an autoimmune disease, in which T cell attack myelin sheath and damage, there are lots of questions that we need to be answered. There are many tolerance mechanisms in body to prevent self-reactivity. We know that central tolerance is not perfect, and we talked about that. There are autoreactive T cells present even in healthy people, but there are still peripheral tolerance which keep these cells quiet. So probably these peripheral mechanisms are broken in MS, as we talked about the differences in regulatory lymphocyte. Another issue here is that CNS is believed to be an immune privileged site without immune cell trapping. So how MS can occur? For a long time, CNS has been considered as an immune privileged site because of different reasons. There is no transplant rejection in CNS. There is no lymphatic vessel in parenchyma, so antigen cannot be transferred to the lymph node. There is a limited expression of MHC molecule, and also because of the blood-brain barrier, entry of immune cells are restricted. So how T cell responses against foreign or self-antigen in the CNS are induced despite of all of these limitations that we talked about? These days, actually, privileged concept is not believed anymore, and CNS is recognized as being able to develop immune responses to both infection and estrial injuries. CNS has immune surveillance, and it can protect itself from pathogens and other damaging agents. Professor Britta Engelhardt from University of Bern actually has done a lot in this field about the immune surveillance and the blood-brain barrier, and has compared the blood-brain barrier to outer and inner barrier walls around the castle, and has explained how the unique two-layer barrier allow immune privilege in the brain but not around the vessels. Actually, healthy CNS is continuously monitored by the resident microglia and also blood-borne immune cells such as macrophage, dendritic cell, and T cell. These cells together work to detect any would disrupt the health. So microglia are the main cells in CNS surveillance, but there are also meningeal, vascular, and colloid-like macrophages, which play an important role. We can show CNS in a specific area, so for immune surveillance of the CNS. But we don't know how they can enter the CNS in spite of the blood-brain barrier and all those limited restrictions that we talked about. There are three different ways for the T cells to enter the CNS. They can enter from the choroid plexus or from the meningeal, the subarachnoid space, or they can use the parenchymal postcapillary, the prevascular space. So, consistent immune surveillance, prevascular, meningeal, and choroid plexus macrophages are in the place, and they are ready to present antigen to T cell phase, to effector T cells when they enter these specific areas, and they can reactivate these T cells. Here in the left, you can see the meningeal macrophages in green, that they are present in these specific area to actually help the immune surveillance of the CNS. On the right, you can see a video after a virus infection, T cells are shown in pink, and you can see T cell infiltration and the interaction with the actual meningeal macrophages in these areas, which these macrophages can reactivate these T cells. But in MS, so we learned that the effector T cells during MS, they can enter the CNS through these specific ways. But still there is a question that how the naive T cells get activated. So what's the initial trigger for the T cell activation in MS? There are two proposed models. The first one is that antigen might be exported from the CNS and presented in the CNS draining lymph node in the left panel, as you can see. But in another model, which is a peripheral model of the MS, CNS reactive T cell might be activated in the periphery by non-CNS antigen through mechanisms such as molecular mimicry or bystander activation that we will talk about them later. Regarding the first model, the left one, I told that there is no lymphatic vessel in parenchyma. So how CNS antigen can be transported and presented in lymph nodes? There is some recent evidence showing that CNS is directly connected to secondary cervical lymph nodes. For example, if we administer antigen in the ventricle, this antigen can be found in the cervical lymph node 2 hours later. So CSF has a connection with lymph node, and it has been shown. And interstitial fluid contribute to CSF formation. However, its contribution is low, but it's good enough that soluble parenchymal CNS antigen can access to CNS. So after they are in CNS, they can be transported to the nasal mucosa and then to the deep cervical lymph node. So it has been assumed that in some specific situation, such as inflammation, phagocytic activity of microglia are reduced. So there are lots of myelin exosome in the CNS that can access CSF and then transported to the cervical lymph node. So initial activation can occur there, and T cell priming may occur in this place by CNS antigen, and the different types of lymphocyte can get activated in these lymph nodes. And this model is more in line with the intrinsic hypothesis. These cells can enter the CNS through meningeal or choroid plexus port. So we should also mention the role of antigen molecule on effector cells and also chemokine and chemokine receptor during this migration. When they enter the subarachnoid space, they get reactivated, and they are able to produce some cytokines, and these cytokines are able to activate the post-capillary venules and make these venules to become more permissive for other T cells to enter. So local blood-brain barrier disruption and damage result in huge influx of lymphocyte and monocyte, which finally increase the blood-brain permeability in distal area as well. And we will have lots of the tissue damage during the disease. But in the peripheral model, CNS-specific autoreactive lymphocyte can be activated in periphery by molecular mimicry, which means that there are some antigen in peripheral that are similar to CNS antigen. For example, some virus antigens or some smoke constituents and or microbiome contribute to this process. Also, maybe there are some sequestered CNS antigen which are released in the periphery, or maybe there are some inflammation which activate the also autoreactive T cells, which we call it the bystander activation. So environmental factors are contributed to this process, So when lymphocytes get activated in peripheral lymph nodes, they can migrate through the same ways we talked about to the CNS, and that actually result in damage. If we block the migration of pathogenic lymphocytes, we are able to decrease the disease activity. So, and there are some therapies in this regard. And this peripheral model is consistent with EAE. Here you can see the pathogenic cascade. In the EAE, we inject the antigens and adjuvant, and the activation happens in the peripheral lymph nodes. And then these activated T cells and B cells can go to the CNS through the same ways that we talked about. So in this slide, we can summarize early stage events that happens in the early stage of MS. So effector autoreactive T cells and B cells enter the CNS through the subarachnoid spaces and also prevascular space. There are lots of inflammation and infiltration during the early stage of the disease. And I think the prevascular accumulation of cells in prevasculars are the main features of the early stage of the disease. And the tissue damage or axonal damage, as you can see on the right, are mainly caused by immune-dependent ways. It happens because of the cytokine secreted factors, and also it may be caused by the direct interaction of T cells with axons. But during the late stage, immune cell infiltration decrease. As you can see here, there is not that much of inflammation and infiltration. It's maybe because of the lymphocyte exhaustion, but still, chronic intrinsic inflammation and tissue damage continue. One important feature of late stage is presence of the organized inflammation in meninges with increased B cell proportion. These structure in progressive MS allow B cell act independent of peripheral. In progressive MS, gray matter lesions are more abundant. Sorry. Okay. In the progressive MS, gray matter lesions are more abundant and more than 60% of cortex. Meningeal inflammation seems to be an important cause of cortical demyelination by cytokine diffusion. There is more demyelination, but less inflammation during latest stage of the MS. Macroglia and astrocyte activation are main feature of latest stages, and tissue injury is caused by immune-independent mechanisms, which are important during progressive MS, such as acute or chronic oxidative stress, okay, sorry, I just get some message from my computer. I thought that I got disconnected. And so the chronic, acute or chronic oxidative stress, mitochondrial dysfunction, increase of the iron or hypoxia or increase of glutamate. These are all non-immunologic processes can cause the tissue damage during the progressive MS. But still, I want to mention that macroglia activation and activated form of astrocyte or A1 astrocyte are main feature of during the progressive. And also we can see some organized inflammation with lots of B cell in the meninges. So we can end this seminar with some key points. The etiology of multiple sclerosis is unknown, but many features of but many findings indicate a central role for immune system in the disease. Traditionally, MS has been considered to be a T-cell mediated disease, but the importance of the B cells in the pathogenesis of MS is beginning to be appreciated. There are important differences between the pathology of MS in human and EAE in animal models, particularly with regards to the involvement of the B cells and CD8 positive T cells. Targeting of the immune response is most widely used treatment approach for MS, but they are also effective in reducing only effective in reducing relapses with no effect on disease progression. And it might be because of different underlying mechanisms for early stages and late stages. Yeah, thank you so much for your attention and joining me. So I would be happy to answer any questions.
Great, thank you, Samira.
Thank you. Sorry if during the process, sometimes I got some messages that I got disconnected, so I wasn't sure that are you still hearing me or some other things.
No, it was all good. So when you stop sharing your slide, you may be able to see the chat section.
Okay.
We have one question so far.
So apart from the T cell differences, types of immune cells are able also involved in MS. Do we know if particular cells infiltrating or activated in CNS of different patients? Do we see the change in the landscape of the immune cells? So the first question is made about the particular cell infiltration. Sorry, let me just open the chat box here. So Manoj asked about the specific T cell infiltrating to the CNS in different patients. Yes, there are different, so MS is a heterogeneous disease. We can see different symptoms. Different immune responses. So that's the need for the personalized immunotherapy, because antibody and B cells are more important in some patients, but other patients show more CD8 or CD4 T cells. So I think that MS is so heterogeneous, so different. There are different patterns seen between different patients. And next question, in relapsing remitting MS, are 3 certain immune subtypes known to drive relapse? What does the immune system look like during the remission? So it has been shown that during the remission, more actually phenotype of the immune cells switch through the regulatory phenotypes. There are more regulatory T cells, and also there are more T helper 2 cells. These are the evidence that we know. And also the immunosuppressive processes are more abundant during the relapses. And all the regulatory mechanisms body work together to suppress inflammation and immune responses during the relapse. So they are successful to decreasing the inflammation, and also the repair happens during the relapse. But because of the epitope spreading and the new antigen that are exposed now because of the tissue damage, the other relapses will be happen later. And Dr. Young asked that then you show the increasing complexity. So the antigen presentation within the CNS, are there other mechanisms in the CNS that further activate T cells, such as TL-17? So exactly. And when the disease develops, there are lots of factors influencing the disease. We have a different microenvironment in the CNS that they can affect the T cells. For example, we have lots of the extracellular matrix in the MS lesions and in the EA lesions. So, and there are some evidence showing the inflammatory role of the extracellular matrix, so they can affect the T cells inside the lesion. And we talked about the plasticity. The T cells can switch to regulatory phenotype or pathogenic phenotype. So the environment inside the lesion can affect the disease process through affecting the switching the T cell activation status. Are there other ways to prevent the infiltration of immune cells into the CNS besides the blood-brain barrier? So there are some recent there are actually some available therapies such as natalizumab that they can, it is the antibody against the alpha-4 beta-1 integrin on the surface of the T cells. So they can inhibit the migration of T cells to the CNS. And also there are other therapies such as fingolimod. They can actually keep the T cells inside the peripheral lymph node. And both of them are so successful in the MS. So if we block the migration, we can see the recovery. But I am not aware of other methods to prevent the infiltration of the immune cells into the CNS. These are the two important drugs that we have used. You missed the, oh, sorry, what's the Maggie's question? Oh, sorry. Yeah, somebody asked about the vitamin D. Oh, there are some reports showing the vitamin D deficiency affect the multiple sclerosis. Yeah, because the vitamin D can affect the development of regulatory T cells through the signaling that they can activate. So it has been speculated that vitamin D deficiency can decrease the regulatory phenotype of T cells. And in this case, they can contribute to the development of the MS. Can you comment on different types of myeloid cells in brain during MS and their contribution to lesion development? Yeah, there are different kinds. So we have microglia, which are the resident macrophage of the CNS, but during the MS, there are lots of myeloid cells that are coming from the periphery. And the two class, there is a historical concept that myeloid cells can be M1-like or M2-like cells. So M1-like cells, they are more inflammatory. They secrete inflammatory cytokines, but M2-like cells are more protective and produce some anti-inflammatory cytokines. But also dendritic cells among the myeloid cells are present in the prevascular infiltrate and also in dendritic cell and the MS. Good to have question from Megan during final. Others question, do we also see neutrophil activating during different stages? Yeah, during the earliest stage of the MS, we see expansion of neutrophil, as we talked about that. And also during the acute phase of EAE, in both CNS and CSF, we can see increase in the neutrophil. But the role of neutrophil is not clear. Probably they are helping the antigen presenting, and also they are producing a reactive oxygen species, and they can actually enhance the disease development in this way. And when we deplete the neutrophil, we can see EAE. So it seems that they have an important role during the disease development, but the specific role is not clear yet. I think, yeah, any other question?
Yeah, one more.
Oh. Is there any way for the progression of MS to be predicted based on the antigen antibody? 1%. So you're talking about the predictions based on the antigens? I don't think so, but there are lots of other factors that can help us to anticipate the relapses. For example, looking at the inflammatory markers or immune cells in the periphery will be helpful, but I'm not sure about the antibodies, because most of the antibodies are present in MS are not specific. They are for the ubiquitously presented antigens in the body. They are not really very specific to the CNS. That's to my knowledge.
So,
is there any way? Oops.
Yeah, that's the same question. Yeah. So thank you so much, Samira.
Thank you.
It was a great talk. And everyone, thank you for joining us. And we'll see you next Thursday.
See you.
Bye.
Bye.